Hello everyone! In today's lab we're going to be looking into biological databases. Now there's quite a lot of databases out there, so we can't go through them all, but we can go through a few of them and a few rather useful ones, and this will be part of building your toolbox of useful things to have on hand. So databases, very useful when you're doing bioinformatics, but even if you're not doing bioinformatics, these databases are useful places to look up information for the ordinary biochemist anywhere. So databases are in general repositories of information. It's stored and organized, and we can search it to retrieve information that we're interested in. There are a few different types of databases. Loosely, we can say that there are the primary databases where the data is submitted by users, and there's typically very little processing being done apart from what the user had done before submitting. Then there are the secondary databases, which are secondary in the sense that they are using data from primary databases, but something additional has been done to them. And this can be something like annotation, which could be either computational or manual. There could be a curation step or some sort of processing on the primary data and then storing the processed version. There are, of course, a large set of various specialized databases. So these are more focused databases and catering to a more limited niche. But odds are, whatever your niche is, there's a specialized database out there for you, or maybe a number of them. There are also portals, which are sort of sites that allow you to do a search and retrieves data from multiple different databases. So it's a nice shortcut for accessing many different databases with a single search. It is with a portal that we start off our introduction to this lab, namely the Entrez portal, which retrieves data from the NCBI databases. And here I'd like to start talking a little bit about searching these databases, because that's going to be a bit of a theme. And typically we can do rather advanced searches, and at the core of this are the logical operators and or not. So using these, we can be rather specific in what sort of combinations of search terms that we want to put together and find. So let's look a little bit at one results page. So this is a result page having chosen the structured database out of all my hits from searching Entrez. Here, I'd like to draw your attention to the search detail box. This one, on some of the databases, when accessed through Entrez, you'll have this sort of interface. Some will look a bit different, but when this is the case, the search detail box is available, and it will tell you how the database actually interpreted the search you did. So I search for NACHR, so that's the abbreviation for nicotinic acetylcholine receptors, and Homo sapiens organisms. So I would expect it to find something related to the nicotinic acetylcholine receptors, and the organism for it should be humans. So it's the logical and, so for the search to be, for the record to be a hit, it would have to contain both of these terms. And the search detail box is showing that even though I didn't specify all fields in my search, having specified no field, it interpreted that as any field or any type of information is a go. Whilst for searching for Homo sapiens as organism, it added the quotation marks, which means that it has to be exactly like this. Now, the search details, it's useful to see how what you search for is interpreted, and when you're used to it, you can look at it, and if you know how to type something that the portal would understand, you could type it straight up in the ordinary search box, or you could edit things here in the search details and launch a new search. When you're starting off, you're not going to know what all of the accepted fields are, and that's where the advanced search page is really very useful and more human-friendly. So you can select what field you want to search for your term in, and you can choose what logical operator that you want to use to string these different terms together. Now, the next database that we're going to bring up is the PublMed database. So this is a database of publications. Now, there are both the PubMed Central database and the PubMed database. Both are databases of publications, but with PubMed Central, you can access freely available full-text articles, whilst PubMed stores mainly the abstracts, and you will typically have a link to the full-text article, and that will link you to either PubMed Central or to the actual journal itself and the article there. The search options you have available to you are really quite similar to what we looked at with Entrez and that example, but the interface looks slightly different. But you still have the use of your Boolean operators, and you have a button with advanced that allows you to build a more advanced search. Here it looks a little bit different, but you can pick what field you want to be searching for and what your term is, and then you press what logical operator you want, and it will add it to the query box. So if I were to press AND, it would string these two things together, and I would end up with, well, essentially what's search number 2 here in my search history. And the search history and search details is sort of the search detail box from the last slide. So we can normally just see what our search was. So search 1, I haven't expanded the details, so we can see that I just searched for the acetylcholine receptor. Expanding the details, we get what was the contents of the search detail box, so how it interpreted my search for acetylcholine receptor and Unwin as an author. So this can be quite informative and allow you to build more powerful searches and get familiar with how the interface works. Once you do get to the page with actual result, you end up with having a list of the various different publications that matched the search you did. And then on the left of the page, you'll have access to a number of filters, so you can filter down the hits to things that you are the most interested in looking at. Next up, we have UniProt, and this is a secondary database in that it's combining a number of different databases, and you can look at all of the lovely results. You could do an argument that it's a bit of a portal in and of itself, but in general, UniProt is one of the most useful databases out there if you want to look into what's going on with a certain protein. So it has a little bit of information about what the protein does, it has the sequence, it has a little bit of information on like the topology, if there are structures available, it will have links to those. So it's a great place to start off with when you're looking into a new protein. Now, the database itself, it's sort of a conglomeration of a number of them. So we have a few, I think they're primary databases, EMBL, GeneBank, and the DDBJ, so it's Data Bank of Japan. So it's all three of them are nucleotide sequence databases. Then we have a secondary database, Tremble, which is a translation of these three nucleotide sequence databases. These three actually sync their data with them like every day. And then if we translate it to protein sequence, then we have the Tremble database. Add manual annotation, you get SwissProt. And the hits you get in UniProt, it will tell you with these little symbols whether it's a record that they have from the SwissProt database, and these are typically the better because manual annotation, someone's actually looked at it. But there will also be hits from Tremble, and well, it takes a while to look at all of the various proteins out there, so it may be useful to look at the Tremble hit if you don't have an annotated hit. So later on in the course, when you start working on your secret sequence, we'll talk more about that at some point, but also start in time. UniProt is a great launching point for looking into your protein and using it as a database with information and something that most likely links you on to more information that's going to be useful for you. Now, we also have one database that if you're dealing with protein structure is going to be very, very useful, and that's the Protein Data Bank. And this is a repository of experimentally determined three-dimensional structures of biomolecules. So this is where you go if you want to find whether there's a structure of your favorite protein. Of course, there's advanced searches here, so you can find, say, if it has a certain ligand, or you can search for similar things. This interface looks a little bit different than what we've looked at so far, but it's still something that allows you a lot of power over your search. Now, to contrast with PDB, where we have experimentally determined structures, there's also the AlphaFold protein structure database, and this is a database of predicted structures. So the AlphaFold 2 algorithm takes an amino acid sequence, and then it does a prediction of what it thinks that this sequence most likely would look like as a 3D structure. And the algorithm that is doing the prediction, it's based on deep learning and bioinformatics. So it's one of these things that we're going to return to in more depth towards the end of the course. Now, zooming out a little bit from the single protein level, there are databases that have a little bit more of an eagle-eyed view on things. And here I would like to mention KEG. So it's a database that's integrating a lot of different types of information. So from genetic information to chemical to like biological systems function. And they're very interlinked, the various records between them. Specifically, I'd like to point you towards the K-pathways section of it that I find to be fun to look at. And this is essentially a collection of various maps that are showing various different functions in biology. So I think here we're looking at a synapse. So it shows you a little bit about what proteins are doing what in different pathways. So you have the enzyme number, you have what product it puts out and what substrate it takes in, and you can click on all of these things and get a nice easy access to information about either the protein or its substrate or its product, and it's really quite neat and fun to just explore. Finally, we're going to be talking about the GO database, or the Gene Ontology database. And ontology is the philosophical study of being. So what they're trying to do with this database is to unify terminology and define concepts and their names, and how they relate to each other. So it's maybe a bit fuzzy at first, but in practice, what it does, it's describing proteins or other gene products by three main categories. So the molecular function, what does it do? Cellular component, where is it or where is it doing its thing? And biological process, so what pathway or more larger scale process is this thing a part of making happen? And with that, I think that's the introduction I was planning to give you on biological databases. So have fun exploring them and doing the quiz.